Good morning. In this video, we are going to understand more about the mechanism of action of the inhibitory protein P53 that has an inhibitory effect on the cell cycle, which is exerted only when P53 detects the presence of an abnormality in the DNA of the cell. So P53 is a negative regulator of the cell cycle. And that's why it is called a tumor suppressor. Why is it called a tumor suppressor? Because it inhibits the cell cycle when the cells are mutated. It inhibits the cell cycle when there is a DNA abnormality that is detected in the cell. P53 is not the only inhibitor of the cell cycle, but there is another inhibitor of the cell cycle that is less famous, that is called RB or retinoblastoma protein. And this is another tumor suppressor protein that is capable of stopping the cell from dividing too fast. And this is important when the cell has an abnormality. When the cell has an abnormality, like a DNA abnormality, the cell is arrested altogether and enters in the G note, where it is prevented, whether temporarily or permanently, from re-entering the cell cycle. But stopping the cell from dividing too fast is the ordinary control mechanism that is done by P53, even with the normal cells, because even the normal cells shouldn't be allowed to divide too fast. So when is the cell arrested? The cell is arrested when something is wrong with the cell or when certain conditions are not met. For example, when there isn't enough nutrition or oxygen for the cell. So whenever we have, for example, a DNA damage that is capable of causing a mutation in the cell, or a DNA replication that wasn't complete during the S phase, or any other issue that is serious and that we don't want to transfer to the coming daughter cells, then the cell should be arrested. These regulators that slow down the cell cycle or even completely arrest the cell cycle are called negative regulators of the cell cycle. And these negative regulators of the cell cycle have as a major task to make sure that the cell does not proceed to the next phase until the appropriate conditions are fulfilled in order to prevent transfer of errors of DNA or transfer any damage in the cell to the coming generations of cells or daughter cells. So the negative regulators are important because when they arrest the mutated cells, then they prevent the abnormal cells, like the cancer cells, from what they are characterized by, which is the uncontrolled cell division. We all know that one of the major issues with the mutated cancer cells is that they have an uncontrollable, very rapid rate of cell division. So the negative regulators, by discarding the mutated cells, by arresting them, by forcing them to go to the G0 phase, or by arresting them, they prevent them from doing their favorite thing, which is the uncontrolled, very fast rate of cell division. On the other hand, we have positive cell cycle regulators, which we talked about in the previous videos, and we can refer to those videos to review the information like the cyclins and the CDKs or the cyclin-dependent kinase. The positive regulators, on the other hand, ensure that the cell cycle progresses with a good pace that is enough or that is adequate for the needs of the cell. So P53, which we are focusing on today, is a crucial tumor suppressor protein. Generally speaking, P53 is an inhibitory protein, meaning that it will check on the cells at the G1 point or the restriction point that lies between G1 and S. And if it finds any abnormality, it will pause the cell temporarily in an attempt to correct the DNA abnormality, or permanently if the damage in the DNA proves to be irreparable. And because it stops and arrests those cells that carry mutated DNA until otherwise mentioned, then it prevents those mutated cells from dividing in an uncontrollable manner and making a tumor, and that's why we call it a tumor suppressor protein. So how does P53 act as a tumor suppressor protein? By suppressing or inhibiting the cells that have DNA damage and that are liable to divide in an uncontrollable way and produce thousands of cells, of mutated cells like them, mutated daughter cells that will form a mass of mutated daughter cells and therefore a tumor. And that's why through this negative regulation of the cell cycle, it is protecting the cell against the spread and the propagation of the mutated cells. And in this video, we are going to focus more on the detailed mechanisms that are carried out by P53 to achieve those tasks. Number one, P53 will assess the DNA quality of the cell. That's to say, as you can see you can watch the details of this in the previous video that was talking about cancer cells and the relation to P53. So P53 checks for DNA damage during the cell cycle, especially at the G1 checkpoint. So actually, P53 is helped, as we said in the previous video, by a sensor protein, and that sensor protein binds with DNA and detects the DNA abnormality and activates the P53 to bind with the damaged DNA and activate an inhibitory protein. We can refer again to the diagram that we used in the previous video to remind you of what we said about the role of P53 in halting the cell cycle and arresting it in case we have a mutation in DNA. Here you can see that there is a DNA damage, and the DNA damage is sensed by a sensor protein. The sensor protein will activate the P53, and the activated P53 will bind to the damaged area of DNA and will thus activate an inhibitor, and that protein inhibitor inhibits what? It inhibits the cyclin-dependent kinase, in this complex cyclin-dependent kinase. When it binds with this complex, it inhibits, as you see the cross here, it inhibits cell cycle progression. So it will stop the cell cycle at the G1 checkpoint in order to buy time for repair of the DNA. And if the DNA damage proves to be irreparable and the repair proves to be impossible, then there will be triggering by P53 of apoptosis. But if P53 itself is mutated in the cancer cell, then even when the damage of DNA is sensed by the sensor protein, P53, because it is mutated, it will not be capable of binding with the damaged DNA and therefore will not be capable of activating that inhibitory baby blue protein that we saw in the diagram above, and therefore there will be no inhibition of the cell cycle. And therefore, all these functions that were supposed to be done by P53, like stopping the cell at the G1 checkpoint to have time to repair the DNA or sending the cell to apoptosis, all this will not be done, and the cell with unfixed damage of DNA will escape the checkpoint because it is mutated and ineffective, and therefore the cancer cells will pursue their journey and will continue their cell division. And that's why a mutated P53 is a very serious condition for the cell because it allows the cancer cells to continue. So, assessment of DNA quality, this paragraph is what I've just explained on the diagram. So, P53 does not sense the presence of damaged DNA, but it is a sensor protein, as we said. But once P53 is bonded, once the sensor protein detects the damage, it will activate P53. P53 will stop the cell cycle because it wants a repair process to be activated. So the overall action is called assessment of DNA quality, or checking whether there is DNA damage or not during the cell cycle. But once again, I say that, and I emphasize that the checking is not a function of P53 only, but before P53, we have a sensor protein. And if damage is detected by the sensor protein, P53 will be activated, and when P53 is activated, it will initiate a repair process or halt the cell cycle. Actually, in both cases, there will be a halting of the cell cycle. So even when we want to repair DNA, we need to stop the cell cycle first in order to start the repair process, and therefore allow only intact DNA to be used by the cells, or to allow only cells with intact DNA to be capable to undergo mitosis and give rise to similar or identical daughter cells. So the first attempt is to stop the cell cycle in order to repair the DNA. But if the DNA is irreparable or proves to be irreparable, then in such a condition, the P53 will trigger or will initiate apoptosis. What is apoptosis? Apoptosis is programmed cell death. So apoptosis is a very regulated series of processes that result finally in a very clean and neat death of the cell, where the components of cell damage or cell death will be recycled and will not result in any inflammation. So apoptosis is a process that is very organized, That's why we say it is programmed cell death, because it's a very organized process of cell death in which the certain enzymes help in breaking down the cell components, but at the same time, the results of breaking down of the cell will be like put in or engulfed by macrophages, so that there will be clean recycling of such components, and therefore there will be no inflammation. So it differs from degeneration. So if the DNA damage is irreparable, if the DNA damage is too severe, then the cell will stay permanently in the genote, as we said in the previous video, and you can refer to apoptosis again in the video that has been uploaded previously on the channel to know the details of apoptosis. So if the DNA damage is too severe and cannot be repaired, so P53 will activate and trigger certain processes to start apoptosis. What is apoptosis? It is programmed cell death. It is a kind of cell death that does not initiate inflammation and where all the components of the cells can be recycled once again, and that is necessary in case the cell has any kind of damage. So apoptosis in such a condition will prevent the mutated cell from proliferation. So the importance of apoptosis in case of damage of DNA is that it prevents the occurrence of cancer, because it prevents the mutated cells from proliferation. The third mechanism of action by which P53 acts is to recruit repair enzymes. We said that when P53 detects the presence of damage of DNA, it arrests the cell in G naught. And this time during which the cell is arrested is first used to try to repair DNA. And if DNA is irreparable, the cell goes to apoptosis. But this means what? This means that we need repair enzymes. Which molecule will be used to activate those repair enzymes when repair is possible? It is P53. So one of the major functions of P53 is to recruit and activate the repair enzymes, and therefore the chemical pathways of repair of DNA to fix the damaged DNA. Needless to say that if P53 itself is mutated, because in cancer cells, the problem is that we're not going to hire a normal P53 from another cell. P53 is present inside the cell and is a protein that is expressed by the genes of the cell. So if you have a cancer cell with mutated DNA, so it would be extremely difficult for the P53 of that cell to be normal, because most of the time it will be as mutated as the rest of the genes in the cell. And this is the cause behind the success of the malignant cells in escaping the expected checkpoint P53, because that P53 checkpoint itself is not functioning properly since the gene that expresses it is mutated. So if the cell is cancerous because of mutated DNA, so most of the time the P53 itself is faulty or mutated, and therefore all those regulatory mechanisms that we are expecting to protect us from cancer will themselves be mutated. And this is what allows the cells that have damaged DNA, like the cancerous cells, to continue divide and lead to cancer. As we said before, if the repair is impossible and if the cell's DNA is severely damaged in a way that prevents repair or makes repair impossible, then P53 will take the other option, supposing that, of course, we're talking about, again, I emphasize once again that we are talking about the role of P53 in cells with damaged DNA, but where P53 itself is not mutated. Because if P53 itself undergoes mutation together with the other genes of the cell, then there is no hope that this P53 could be able of correcting the abnormalities of DNA in the cell. So we are talking about a cell that has DNA but damage, but whose DNA damage does not affect the P53. So in such a case, the P53 is still functioning. It can either cause repair of DNA if possible, and if impossible, it will trigger apoptosis and it will induce cell death. In such a way, the P53 will prevent the cell from continuing to divide, because if it divides with all these mutations of DNA, it will transfer those mutations to the daughter cells, and this is what we don't want to see inside a living cell. Why? Because if a cell has faulty DNA, damaged DNA, then it will produce faulty proteins, because damaged genes or mutated genes cannot produce normal proteins. And if you have faulty proteins, this simply means that you have faulty enzymes, that you have faulty biochemical pathways, that all the normal cellular functions will be disrupted, leading to diseases, starting from mild diseases to severe diseases like cancer. On the other hand, promoting apoptosis is a protective mechanism to the body. So apoptosis is not, although it is called cell death, but it is programmed cell death whose aim is to protect the body again by discarding the cancerous cells or the mutated cells that, if are allowed to reproduce and divide, they will cause tumors, malignant tumors in particular. So apoptosis will benefit the organism. How does apoptosis benefit the organism? By preventing the cell division of damaged cells, by preventing the proliferation of damaged cells, and therefore by maintaining the integrity of the cell. When you discard mutant cells with mutant DNA and keep only normal DNA that codes for normal proteins, then you are maintaining the integrity of the cell. So we can say that P53, by being a tumor suppressor protein that regulates the cell cycle, is important for maintaining genomic stability. What do we mean by maintaining genomic stability? We mean that it discards all mutated DNA and keeps only the normal DNA that codes for normal proteins that will carry out normal functions in the body. Again, we are summarizing the functions of P53. Whenever there is DNA damage, then the P53 will induce cell cycle arrest. And when is this done? Either at the G1 restriction point or at the G2 restriction point. The G1 restriction point lies at the end of G1, growth phase 1, and before the start of the synthesis phase or the phase of replication of DNA, whereas the G2 restriction point occurs at the end of the synthesis phase and makes sure that DNA replication has been done correctly. When the cell cycle is halted or paused after G1 or even after G2 and before entering into, the G2 here, I mean after the S phase. So whether it is after the G1 phase, which is the G1 restriction point, or after the S phase, which is the G2 restriction point, the aim of such arrest is not to discard the cell for no reason, but it is to buy time to try to repair the DNA of the cell before proceeding with cell division. How can we stop the cell cycle to buy time for repair of DNA? Here is a more detailed piece of information. We always said that P53 buys time by stopping the cell cycle to repair DNA, but we have never said how P53 can do this function of arresting the cell cycle either temporarily or permanently. It does this by activating a protein called P21. And this P21 is an inhibitor of the CDKs or of the cyclin-dependent kinases. And since the CDKs or the cyclin-dependent kinases are stimulators to the cell cycle, then inhibiting them will stop the cell cycle. So the additional piece of information that we have learned from that paragraph is that P53 stops the cell cycle by doing what? By activating an inhibitory protein called P21 that activates a cyclin-dependent kinase. And again, as we said, the temporary arrest of the cell cycle is important for DNA repair. How does P53 help in activating the DNA repair? P53 does a very important function, which is to activate the expression of DNA repair genes. There are genes in the cell that are called DNA repair genes. So when P53 stimulates their expression, this means that it stimulates their translation into proteins, and those proteins are nothing but enzymes of repair of DNA. And this is the way P53 helps activating the DNA repair mechanisms. So once again, the pause that P53 does with the help of P21 is important for having time to activate the genes of repair. What are the genes of repair? They are genes that, when expressed, they give you enzymes that help and proteins that help in repairing DNA. And therefore, if the damage is actually repairable, then the cell will return to the cell cycle. But of course, here we have one important condition, that the DNA repair genes themselves should not be mutated with the rest of the mutated genes in the cell. The third function is apoptosis. I know we have mentioned it earlier in the video, but now again we are repeating this to add a new piece of information. We said before that if the damage in DNA is an irreparable damage, if it proves to be an irreparable damage, then the cell will be forced to go to programmed cell death or apoptosis. This occurs when the DNA damage is extensive in the cell, when it is very severe in the cell, or when the DNA damage proves to be irreparable in the cell. That's to say, the enzymes of repair tried to repair it but failed. In such a case, P53 will initiate the process of apoptosis. And the importance of this paragraph is that, that it shows us how this initiation is done. This initiation is done by the fact that P53 will activate certain proteins that are known as the pro-apoptotic proteins. What do the pro-apoptotic proteins do when they are activated? They cause disruption of the mitochondrial membrane. That's to say, they cause rupture of the mitochondrial membrane and release of certain proteins that will trigger a cascade of reactions. And this cascade of chemical reactions will finally lead to cell death in a very organized way, in a very neat way, in which the products of lysis or hydrolysis of the cell will be engulfed by the macrophage in order to avoid any inflammation in the cell. So in such a case, as we said before several times, this is a protective mechanism that protects the cell against cancer. So when apoptosis occurs, it is as if some cells in the body sacrifice themselves for the benefit of the whole body. And the fourth mechanism by which the P53 fights cancer cells is what we call senescence. And senescence means aging. How does this occur? When the cell damage is extensive and when the DNA is irreparable, we can see that P53 can induce cellular senescence or aging. So cellular senescence or aging is induced by P53. So this is a fourth function of P53 that we are mentioning here because we are going into depth more in the actions of P53. What do we mean by senescence? In senescence, the cell will not undergo apoptosis. The cell will stay alive. So the cell stays alive. But what happens is that the cell is arrested from division. So it will stay permanently in the G note phase. So it was not repaired, it did not undergo apoptosis, and it's just arrested, and it will be later on recycled in the body. So overall, P53 plays a key role in protecting the organism from cancer by ensuring that those cells with damaged DNA will never be allowed to proliferate. So cells with damaged DNA cannot be allowed to proliferate. There is one important question that you frequently found in your tests, in your AP tests. How can so many functions be supported by a small molecule like P53 that is a single molecule and that is small in size? So the ability of P53 to support multiple functions, like the four functions that we have just mentioned, is related to the fact that it is a modular protein structure or a modular protein molecule. So what we are going to explain now is related to protein structure and function that we have mentioned in biochemistry, and to cellular signaling that we have mentioned in the videos on the cell. And you can refer to those videos to understand more about what we are explaining now. So there are several functions that help P53, several properties, that allow P53 to carry out all these complicated functions, although it is a single small molecule. The first one is the modular protein structure. What do we mean by a modular molecule or a modular structure? It means that this, of course, that proteins are, in order for proteins to be functional, they should be in the 3D configuration, at least in the three-dimensional structure. And in the three-dimensional structure, they always have specific regions or specific domains that can bind with only specific molecules or signaling molecules. And those domains differ in what or why are they specific? By their shape and by their charge. So those domains on the three-dimensional proteins can only bind with molecules that are complementary in shape and complementary in charge. And these domains allow the P53 to interact with various partners, and those partners are nothing but the signaling molecules. And this allows them to do what they do of repair mechanisms, to act as cell cycle regulators and to stimulate apoptosis. So the fact that they have multiple domains allows them to cause DNA repair and to regulate the cell cycle, to act as cell cycle regulators and to stimulate or activate the pro-apoptotic proteins to induce apoptosis. Here you can see a protein molecule in the 3D structure, and you can see how a molecule in a 3D structure can have several domains. See this domain, that domain 1 that has a complicated structure, domain 2, and domain 3. All these domains, if seen under the electron microscope, can be seen to show specific configurations and specific charges. So even though it is a relatively small protein, but it is a versatile protein, meaning that it has several domains with several different shapes, with several different charges on those domains, and therefore can bind with different signaling molecules. But there is something else that makes the molecule of P53 versatile and therefore capable of doing multiple functions other than the presence of multiple domains, and it is the post-translational modifications. Because when we explained this before in a previous video on molecular biology, when we talked about the regulation of gene expression in eukaryotes, and we said that there are certain different levels, several different levels of regulation of gene expression, or the way the gene is translated into a protein. Some variations come from modifications that occur after translation of the proteins, which we call post-translational modifications. So P53 itself, after being translated from its gene, can undergo one of three processes. The first one is phosphorylation, the second one is acetylation, and the third one is ubiquitination. So there are three processes that P53 can undergo and that can affect its function, which are phosphorylation, acetylation, and ubiquitination. In these modifications will change the conformation of P53. That's to say, they will change the shape of the molecule of P53. And as you know, any change in the 3D conformation of a protein will definitely change the activity of that protein. For example, phosphorylation is important because it helps activation of P53. So by changing phosphorylation changes the conformation of P53, but it changes its conformation in a way that makes it more active. Similar, on the other hand, we have acetylation. Acetylation diminishes the ability of P53. So both of them, phosphorylation and acetylation, will change the conformation of P53. Phosphorylation simply means addition of a phosphate group, whereas acetylation means that we are adding an acetyl group or CH3 group to P53. While phosphorylation causes activation, acetylation will diminish the ability of P53. What about ubiquitination? Ubiquitination means that we are going to attach P53 to a protein called ubiquitin, and this attachment will label the P53 in order for the destruction enzymes to know where P53 is and to destroy it. So ubiquitination is a way of labeling P53 to destroy it. These post-translational modifications allow the same protein to do several functions or to participate in different pathways. On the other hand, we have the transcriptional regulation. What do we mean by transcriptional regulation? P53 can act as a transcription factor. And again, this is an additional function of P53. And when it acts as a transcription factor, this means what? This means that it helps the RNA polymerase to bind to the promoter and start transcribing DNA. And therefore, P53, just like transcription factors, will regulate the process of transcription and therefore will regulate in an indirect way the expression of the genes. And of course, P53 will not be concerned with any gene. It will be concerned only with the genes that are necessary for cell cycle control. So the last function of P53 we are discussing now is its ability to act as a transcription factor, helping transcription of DNA and therefore helping the expression of this DNA into proteins that are important for cell cycle control or for DNA repair or for apoptosis, which are the three functions that P53 does. So again, this function of P53 of being capable of acting as a transcription factor simply means that it will stimulate transcription and therefore translation of proteins, or shortly, it will regulate expression of genes, but not any genes, the genes that P53 is concerned with, which are cell cycle control, the proteins involved in cell cycle control, or in DNA repair, or in apoptosis, and therefore it activates a great number of target proteins that allows it to do a great number of functions. But P53 shows what we call contextual activation, which means that P53 can respond to different stress signals like DNA damage, like hypoxia, for example, and adapt accordingly. And therefore, we can say that P53 is a flexible molecule that reacts in the cell according to the context in which it is exposed. So it can carry out different functions, but every function is carried out in a specific context. So it's true that P53 can carry out different functions, but it selects the suitable function for every context. And this is what we call contextual activation. And in all these conditions, it adapts the function it does according to the needs of the cell. So contextual activation again means that P53 activates certain functions according to the needs of the cell, and therefore P53 shows great flexibility inside the cell. We will stop here and we will continue our discussion of mutations in the cell cycle, genes, and cancer in the next video. Thank you and see you in the next video.